So, this is the other lecture of the pharmacovigilance system, that is identifying early signals of drug-related problems, from Dr. Gloria Shalviri, and she's a great professor in the pharmacovigilance systems. Our objective in these lectures is what is signal, excuse me, how are signals detected, different source for signal detection, different types of signals, quantitative methods for signal detection, and excuse me dearly, I don't have a coronavirus. This is a COPD I have. Quantitative methods for signal detection we should understand. Signal assessments, signal managements, signals in PSUR, periodic safety reports, and some examples. Data assessment in pharmacovigilance. Individual case assessment. Relevance of observation. Quality of documentation. Coding. Causality assessment. Preventability assessment and seriousness assessment. Okay, for individual case assessment, we explained the causality assessment in the other video. We explained the preventability assessment in the other video. And now we want to go through the others. Data assessment in pharmacovigilance number 2 is aggregated assessment and interpretation. In this case, we should do the signal detection and the risk-benefit assessments. For the signal, the WHO definition is reported information on a possible causal relationship between an adverse event and a drug, the relationship being unknown or incompletely documented previously. Reported information on a possible causal relationship between an adverse event and a drug, the relationship being unknown or incompletely documented previously. Okay, the definition of signal. These are the reported information on a possible causal relationship between an adverse event and a drug, the relationship being unknown or incompletely documented previously. Note: a signal is an evaluated association which is considered important to investigate further. A signal is an evaluated association which is considered important to investigate further. A signal may refer to new information on an already known association. A signal may refer to new information on an already known association. And usually more than a single report is required to generate a signal, more than one, depending upon the seriousness of the event and the quality of the information. Okay, I read it again. Reported information on a possible causal relationship between an adverse event and a drug, the relationship being unknown or incompletely documented previously. A signal is an evaluated association which is considered important to investigate further. A signal may refer to new information on an already known association. Usually more than a single report is required to generate a signal, more than single report, depending upon the seriousness of the event and the quality of the information. What is the definition of signal? In this page is explained that the signal is a warning from every source that may be the probability this drug maybe have a dangerous bits that maybe this is unknown with any low probability or I cannot translate it exactly to English, from quantity or from quality, and from the quality is written the number may be a lot, or from the quantity the number may be a lot, and the different information we have. And this is the other definition. That is a warning from any source that maybe have a dangerous probability of the dangerous, and we don't know it, that is unknown. And from the point of the view of the quantity or quality, have a severity, more severity, or have a more frequency. Okay, again, this other language, I should translate it. It's written in the other words that the first symptoms that appears as a side effects or adverse drug reaction that make a hypothesis, and this hypothesis will be approved for the new adverse reaction or new side effects of these medicinal products, and that is unknown, and we should complete the information about this adverse reaction or these side effects for this new adverse reaction that we don't know, and this is unknown about it. This is a new symptoms, that is hypothesis, and we should approve it, that is it a adverse drug reaction or not. This is a hypothesis, and this is type of the signals. And some of the adverse drug reaction in this page explaining some of the adverse drug reaction is a clinical events, new clinical, that's not a new clinical events, that we want to approve it, but these are the old disease that we know it, but because of the new drugs. For example, thromboemboli, due to the contraceptives. We know the thromboemboli, that's an old disease, but because of the new medicine. This is a signal, and this is a new adverse drug reactions because of this medicine, but that was an old disease. Okay, that was a hard job for me to translate, sorry. Okay, now we should go to this graph, what it wants to show us. This is a knowledge of the adverse effects in percentage from 0 to 100 percentage, and we have a time, signal generation, signal strengthening, and we have a signal follow-up. Okay, when we generate a signal, we don't have a lot of knowledge. When we do the signal strengthening, we increase our knowledge. Actually, we do the signal assessment, we have a 50%, 60% knowledge, and after that, when we follow up the signal, we increase our knowledge, and we never touch the 100% because Actually, we are not sure every time the 100% about any medicine. What is the source of the signals? Observations in individual patient, observation in population, or experimental finding? Observation in individual patients, observation in population, or experimental findings. Observation in patients. Qualitative signals, a spontaneous reporting system. For the observation in patients, we have a qualitative signals, we have a spontaneous reporting systems, we have a anecdotal literature reporting, we have a prescriptions event monitoring, we have an intensive hospital monitoring. These are the okay, now we are in the source of signal, and for the observation in individual patients, in observation in the individual's patient, we have a qualitative signals, we have a spontaneous reporting systems, we have anecdotal literature reporting, we have prescription event monitoring, also we have intensive hospital monitoring, such like this. Okay, observation in population. What is the observation in population? If we do the observation in population, we have a quantitative signals, a statistical disproportionality, large spontaneous reporting system. Now we have a large spontaneous reporting system. This is not a small like inpatient. We have a prescription event monitoring again. We have a case control, and we have an intensive hospital monitoring again. For the experimental finding, we have animal toxicology, and we have in vitro experiments. Okay, again review the source of signals or observation in individual patients, observation in population. We have experimental finding, observation in patients. That's the type of signals are qualitative, that is spontaneous. We have a literature reporting, we have a prescription event monitoring, and intensive hospital monitoring of the patients. For the observation in population, that is a quantitative signals or a statistical. We have a large population, we have prescription event monitoring, we have a case control, and we have intensive hospital monitoring. For the experimental finding, we have animal toxicology, and we have in vitro experiments. Factor favoring signal detection. For the factors favoring signal detections, the clinical event, a very low natural frequency, characteristic for unusual signs and symptoms occurring in groups of similar patients, known to be frequently drug-induced, like anaphylaxis, Stevens-Johnson syndromes, and drug exposure high frequency. Okay, again, which factors favoring signal detections? The clinical event, drug exposure, and adverse reactions. These are the factors favoring signal detection. In the adverse reaction, in the drug exposure, and in the clinical event, we detect the signal. For example, in drug exposure, we have a high frequency of the signal detection. In the clinical event, a very low natural frequency we have, but we do the signal detection. Characteristic or unusual signs and symptoms occurring in groups of similar patients, known to be frequently drug-induced, anaphylaxis, Stevens-Johnson syndromes, known to be frequently drug-induced. We should know the frequently drug-induced, and these are explained in the other lectures, and I take a video. In the factors favoring signal detections, adverse reaction, in the adverse reaction also we have a high frequency of signal detections, suggestive time relationship and suggestive dose relationship also, and that is a plausible pharmacological mechanism. We have it in the adverse reaction because we know the drug, we know the side effect, and we have a plausible pharmacological mechanism. Criteria for signal assessment. Which criteria we have? We have a quantitative criteria and we have a qualitative criteria. Okay, for the quantitative criteria, strength of association, number of case reports, statistical disproportionality. For the strength of association, we have in quantitative criteria for signal assessments, number of case reports we should measure, and we have a statistical disproportionality for quantitative signal assessment. For qualitative signal assessment, consistency of data are important. Characteristic feature, pattern, absence of reverse finding. Consistency of data is a qualitative signal assessment. Characteristic feature, pattern, absence of reverse finding are important. If we have a consistency of data, it means characteristic feature, the pattern, and absence of reverse finding. Exposure response relationship, it means the site, timing, dose response relationship, and reversibility. It means exposure response relationship. Biological plausibility, pharmacological and pathological mechanism. Pharmacological and pathological mechanism should be explained in the qualitative criteria for the signal assessments. In the criteria for the signal assessments, qualitatively, experimental finding, rechallenge antibodies, drug concentration, abnormal metabolites. These are the experimental findings. In the analogy, previous experience with drug, often drug induced, are important. Previous experience with drug, often drug induced, are important. Nature and quality of data, objectivity of event, validity of documentation, and causality assessment. Nature and quality of data, objectivity of event, validity of documentation, and causality assessment. Okay. Nature and quality of data, objectivity of event, validity of documentation, and causality assessment. Okay, again, the signal assessments may be quantitative by statistical or number of the case report, strength of association, or maybe qualitative by characteristic feature, pattern, reverse finding, site of exposure, timing of exposure, dose response relationship, exposure response relationship, reversibility of the drug and the body, biological plausibility, pharmacological and pathological mechanism, experimental finding, rechallenge antibodies, drug concentration, abnormal metabolites, analogy, previous experience with the drug, often drug induced. These are the analogy. Nature and quality of data, objective of events, validity of documentation, and causality assessments. The speed of a signal detection. The speed of signal detections depends on number of users of the drug. As many as number of the users of a drug increase, the speed of signal detection are increase. Frequency of adverse reaction. frequency, as many as the frequency of adverse drug reaction increase, signal detection increase. Reporting rate and quality of documentation. If the quality increase, the speed of signal detection increase. Quality of documentation, reporting rate, frequency of adverse reaction, and number of users of the drug increase speed of signal detection. Signal strengthening. Seek information from medical literature, other databases, for example, WHO, the manufacturer, clinical trial records, if available, analogy with other related drugs. Absence of supporting data does not imply false signal. Absence of supporting data does not imply false signal. Signal management. Selection of the relevant data, case reports. Again, for the signal strengthening, if we want to have a strong signal, we should seek the information from medical literature, from database, from the manufacturer, from clinical trial records, from analogy, and the analogy means previous experience with the drug and often drug induced. An absence of supporting data does not imply the false signal. If we don't have absence of supporting data, it doesn't mean that this is a false signal. Maybe I don't have information, maybe there was not a previous information. I should strengthen my signal. Signal management. Selection of the relevant data, case reports, and delineation of the signal, that is a hypothesis. I should manage my signal and selection of the relevant data, case reports, and delineation of the signal, that is a hypothesis. Search in the literature. Gathering of missing data, follow-up of cases, structured inquiry. Search in the drug register files. Assessment and reassessment of all available data, including complete case reports. Selection of the relevant data, case reports, and delineation of the signal, hypothesis. Search in the literature, gathering of missing data. I should gather all of the missed data. Follow up of cases, structure inquiry. Search in the drug register files, assessment and reassessment of all available data, including complete case reports. I should complete all of my case reports, and I should assess and reassess all of my data. I should search in the drug register file. I should gather all of my missed data. For example, in kidney disease, in pregnant disease, in kids, I should search in the literature. In this case, I manage my signal. Oh, where I go? Sorry. Oh, what? Okay. First of all, I should seek the information. Then I should analogy with other related. I should select the relevant. In this case, it means case reports, and I should delineation of the signal. I should make a hypothesis. I should search in the literature, gather data. I should search in the drug register files, assessment and reassessment of all available data. Signal management, writing a report containing summary of the signal, presentation of original data, presentation of additional information, discussion with reference to positive and negative arguments, conclusion, even if preliminary, and suggestion for further study. Okay, how should write a report containing summary of the signal, presentation of original data, and presentation of additional information, discussion with reference to positive and negative argument. I should write a report. I want to manage my signal. In these reports should contain the summary. I should present my original data. I should present my additional information. I should discuss with reference to positive and negative arguments. I should conclude even if preliminary. I should suggest for further study. This is a signal management. The report may serve as basis for decision making by the competent authority and the pharmaceutical company and may be used for the preparation of information for practitioner and in the published literature. This report may be serve as basis for decision making by the competent authority and the pharmaceutical company. It may be used for the preparation of information for practitioner and the published literature. I should write a very good report with the good summary, with the good original data, with the good additional information, discussion, conclusion, and suggestion for further study. I should make a decision. Making decision. Do nothing. Wait for more information. Inform health professional. Initiate further study. Animal study, clinical trials, case control, cohort study. Alter product information. Alter. I should alter and substitute product information maybe. I should make a decision. Maybe I should do nothing. Maybe I should alter. I should inform the health professional. Maybe I should wait for more information. And maybe I should initiate further studies for my medicinal products. Making decision, maybe I should change marketing condition, limit indication, or maybe I should limit availability. A specialist prescription may be needed for the just maybe it should be sold in the pharmacy. Suspend marketing authorization and permanently withdraw marketing authorization. Maybe I should permanently withdraw from marketing authorization or suspend from marketing authorization. I should make a decision for changing marketing condition, limit indication or limit availability. Dissemination of information. What does it mean, dissemination of information? For health professional first, or for pharmaceutical company, or for press release, or for other authorities and WHO? Which dissemination of information I should have? First, health professional, then pharmaceutical companies. In the third is a press release, then other authorities and WHO. It means dissemination of informations. Signal detection methods. Which method we have for signal detection? Denominator-based method, observational epidemiological method, numerator-based method, proportional reporting ratio or PRP, proportional reporting ratio, and Bayesian data mining. Bayesian data mining. Denominator-based method is a cumulative sum, time scan, or Poisson method. Denominator-based method is a cumulative sum, time scan, and Poisson's method. This is a denominator-based method. Denominator, numerator, denominator. Observational epidemiological method. For the observational epidemiological methods, numerator-based method. And this observation, I explain it a lot for the observation in patients, in populations, or experimentals. For the numerator-based method, we have a short memory scheme. Short memory schemes. And while for the denominator, we have a cumulative sum, time scan, or Poisson's method. Bayesian data mining, Bayesian confidence propagation, neural network or BCPNN, and empirical bias screening. Bayesian confidence propagation neural network is a Bayesian data mining, Bayesian confidence propagation neural network or BCPNN, and we have empirical bias screening. Okay.